Okay, good evening, everyone. So I quickly want you to have a look at the question that we had for CAA. So can anybody help me clone this gene here? Okay, before moving ahead, I think it's important that we try to see this first. Yes, can anyone help me? I mean, I have started evaluating and so far I'm midway and I haven't found anyone with the correct answer. So yes, anybody can suggest one? So I would want you to look at, did you have any isocardamers here? So when we clone the gene, right, we wanted it in this way. It should be in the reverse orientation, isn't it? That's what we are looking for. So this should have been the, 1 second, somehow there is a problem. Start and just give me a minute. And this should be the stop codon, right? So how can we clone that? So you should look at any are there any isocardamers here? These two are isocardamers. I mean, this also, all the blunt end cloning ones you can assume isocardamers. Usually it is about it is about the ones that give rise to overhangs, identical or compatible overhangs. I want you to think about this one, MBO and BGL2 as isocardamers. GATC, it cuts here, right? There is a cut here. I can also think of BGL2, AGATCT, and AGATCT. If we cut it using a restriction endonuclease, that is BGL2, it would cut this way, right? This is a overhang. If I cut this, it will give overhang as this. That is the overhang. I hope you can already identify the part that is the actual overhang, GATC, and here too you have a GATC. There are two restriction endonucleases. This one is MBO1 and this one is BGL2. There are different restriction endonucleases. One is a 4-base cutter and the other one is a 6-base cutter. But they would give rise to the same overhangs, GATC and GATC. So the answer for this is that you should have also looked for any restriction endonucleases that give rise to same overhangs. So T is here, and I want to cut. You can use T and BGL2 to cut here. Let me change. So BGL2 and T can be used. And here I could use T and M. So the gene would have been cloned in this particular direction. That would be the answer for that question. Okay. So people have you have tried different things. There is some people have used linkers and some people were stuck in the middle and so on. I did give some marks, but not the full marks for it. Okay. Do you have any questions on this, or if you didn't understand, you can tell me, and then I will try to see if I can explain this differently. All the answer is about looking for isocardamers. 1 second, not T, A. Is there a mistake or anything anywhere? P. Yeah, here is A. A is there, right? 1 second. I'll rephrase it. So the answer is simple. This is you're cutting with BGL2, and here you will cut with you can cut with A or T. Both will yeah, you will cut it with A. Okay, and here you're going to cut this with M, so that the start codon would have been here, and it could be cut with P. P is going to give on the stop codon, sorry, the blunt ends. So this is how you're going to clone it. Okay, that's the answer. I think I got a little confused. Yes. Okay. So we were discussing about analysis of gene expression. You can have a look at this question and get back to me in case if you have any doubts or clarifications required. So in analysis of gene expression, we discussed about Northern blotting, and the focus was that we were trying to discuss about the labeling of the probe. We discussed that there are two kinds of labeling, end labeling, and the other one is random labeling. What we use for Southern blotting is the end blot, the random labeling. So for that, you can use a vector which has two different polymerases like this, T7 and sp6, and provide that particular polymerase and perform in vitro transcription, so that you will get the antisense of it. And then we also discussed about the components that we need to take: buffer, RNA polymerase template, and that at 37 degrees centigrade when you incubate, you will get. So for RNA transcription, you need NTPs. For DNTPs is for PCR. We also discussed about how we could use some of the vectors are made like this in such a way that there are two T7 promoters. So we can cut it with one side so that the other RNA polymerase, see, the promoter could provide us the minus strand. Okay. And we also discussed about how to specifically get the random label. And there we are going to use UTP or any of the bases. Basically, you could have taken any of the bases. We could have taken all the bases as well, NTPs. All of them could have been labeled, but we don't want too much radioactivity. We want adequate amount so that the detection will be sufficient. We don't want to use any extra radioactivity. So, and the other part is we are going to use alpha p32, meaning that why are we taking alpha p32? It is the alpha position is going to get incorporated into the polynucleotide, while the beta and gamma are going to be released out as pyrophosphate, and that will be hydrolyzed by pyrophosphatase or so and into inorganic phosphates. So we would use any of the bases, especially because we are doing it in vitro. That means in vitro, you are giving the template and you are providing everything. So even if you give alpha CTP, for example, alpha p32 CTP, it's all right, no issues. Okay. So before going, I would also like to just maybe I will take up end labeling at a later point. But what I want you to remember is we can use polynucleotide kinase, okay, PNK, which will transfer a phosphate group onto the 5' end like this. So we have to then provide ATP. We will discuss this again sometime else, but right now I remember it as we'll provide substrate for polynucleotide kinase. It's an enzyme, is ATP. The gamma phosphate is going to be transferred onto the 5' end of the DNA or RNA also. So we have to take ATP that is labeled on the gamma phosphate, gamma p32. And just to draw the distinction between what is it about alpha p32 and gamma p32. So here the substrate that we are choosing is, it's only PNK is only going to take the alpha phosphate from here and put it onto the DNA. So we will take ATP that has gamma p32 labeled. Okay, and then it gets here. So N labeling. We will come across these two ways at several points, especially if it is blotting techniques, it's about random labeling. If it is other techniques like primer extension and so on, we would have used N labeling. Why, which one to choose, whether random or N labeling, we will discuss at a later time. Okay. So let's move on with the techniques that we are trying to read. As an alternative to Northern blotting, what is Northern blotting? It is about finding if an RNA sample has a specific sequence or not. And if it is there, how much is it? You can also, by semi-quantitative approaches, you can also, in relation to a control, you can determine if there is more expression or less expression of that particular RNA. So there is another assay that is has similar ways, similar objectives in a way, RNAse protection assay. That means we can produce a template, something like, you can call it as a probe. Okay, so you can produce a minus strand, or you can call it as the antisense strand, and add it to the RNA sample. So you have isolated total RNA, okay, and then you have the DNA, you have made the antisense strand, and you will allow it to bind with it. So our total RNA will have several types of RNAs, including the mRNA of your choice or your interest, and to which the antisense strand you have synthesized. When you add it, the antisense strand will base pair with that RNA. And now we are going to something like this. You have the specific mRNA that you're looking for, and you added the antisense RNA. You allowed the annealing to happen, and then we have treated it with RNAse. So RNAse will degrade all the RNA that is present, except this particular region. So we are choosing an RNAse which is single strand specific. Okay, it can only cleave single strands, not the double strands. So because we have the probe, assume it is a probe that we added, and it is bound, and the mRNA is protected wherever the binding is there, the other parts could have been degraded. And say, for example, with the other ones, they are non-specific. So there is no annealing between them. And when you add RNAs, all of it is degraded. And when you load such sample, you will find one band that is corresponding to this, the molecular weight of this double-stranded RNA. So now look at the technique again. RNAse, the name of the technique, RNAse protection assay. So we are adding RNA such a way that it is providing protection to the RNA of our choice, of our interest, that is this one, right? So that is why it is called as RNAse protection assay. It has some advantages over Northern blotting. So in Northern blotting, or working with any working usually done with RNA is actually a tough one, because RNA is highly sensitive. Can anybody tell me why is RNA sensitive? Anyone, please? Okay, so when we have RNA like this, the ribose, this is CH2OH, and we have OH, we have OH, and here maybe the base and so on. So the 3' OH, this is 1', 2', 3', 4', and 5'. The 3' OH is the one that forms the phosphodiester bond with the succeeding base. In the sense, it would have had the O oxygen bound to the phosphate, and then another the 5' end of the other base here. So the 2' OH, the difference between DNA deoxyribose and ribose is, in deoxyribose, the similar thing, you assume that it is with the another base and so on. Here we have 2' H, that is the deoxy. The oxygen is missing here. So compare it with the one that I'm trying to discuss. So when the RNA is present in alkaline conditions, where it is likely to lose the oxygen, and then that would result in an oxyanion, the oxyanion, which may make a nucleophilic attack with the phosphate backbone here, because of which the degradation of the RNA will happen very quickly. Okay. So a long stretch of RNA, that is one of the reasons why we call it as RNA as fragile, because in conditions there is always the risk that the 2' OH could get activated, and the oxyanion can form, and then it could start breaking the RNA degradation. I think I also discussed about what is meant by denaturation and what is meant by degradation, right? In denaturation is breaking of the hydrogen bonds, whereas degradation is the breaking of the phosphate backbone. So that is one of the reasons why RNA is very fragile, and it's difficult to work with RNA. So when we are trying to isolate and perform something like Northern blotting, right? When we try to do Northern blotting, there is always a possibility that RNA that we have selected is getting degraded, and we might get shorter bands, the bands that are running faster, or fragmentation of mRNA could happen. So this is one good advantage, is that even if it is fragmented, we wouldn't have a problem as long as this particular region is safe. And that's one of the biggest advantages of RNase protection assay over Northern blotting. Otherwise, both the techniques are meant for the same purpose, to find out if an RNA is present or not in a given sample, given RNA sample. Then we have other techniques like reverse transcription PCR. This is simple, where we have the mRNA. mRNA is allowed to bind with a primer. We would have added a primer, and then add reverse transcriptase. And the reverse transcriptase will synthesize. What is reverse transcriptase to start with? It is a RNA dependent DNA polymerase. RNA dependent DNA polymerase. So it will synthesize a DNA strand, that is cDNA, or complementary DNA. Now we can add a second primer, which will bind to the first cDNA strand. We would call this as the first cDNA strand. And upon addition of the second PCR, second primer, we will get the second cDNA strand as well. Now we have a double-stranded cDNA. And we can or we can do PCR with primer 1 and 2, and we can get a large quantity of the product. So this particular technique can be used for, say, if somebody has given us an RNA sample, the question they ask is, we should tell me if a gene mRNA for, I will just write, mRNA for gene X is present or not. And then we would have, if we have known the sequence of the gene X, then we could have designed the first primer for gene X, which should be the sequence of antisense, right? And then, say, for example, if somebody has given us 2 samples of RNA, okay, something like that, or maybe I'll do it It will be easy if we can draw these gel diagrams like this. So if somebody has given you RNA sample 1, sample 2, sample 3, and you are supposed to perform, find out, the only question they would have asked is, tell us if mRNA is present or not. Okay. In that case, we could perform this reverse transcription PCR, and we will run it on the agarose gel. If there is a band, which would have happened only if if only the presence of the mRNA is present. That is when the the primer would bind to start with. Then the first cDNA strand will be synthesized, then the second, and here we have a product. And that product will show up as a signal. If there was no mRNA for that, for gene X, for example, primer 1 would not bind. Therefore, we will not have any reverse transcription, and then we will not have any product in at the end. So we will find something like this. All you can say from RT-PCR is you can say whether RNA is present or not, and if that particular RNA is present or not. Nothing else can be said. We cannot say how many mRNA copies are present for gene X. How many copies of mRNA of gene X are present? We cannot say of 1 and 3 samples. All you can say is yes or no, present or absent. And an extension to this is the technique called as real-time PCR. Many times they would still write it as RT-PCR, and it is confusing because some people take it as reverse transcriptase and some people take it as real-time PCR. You can also call it as qPCR, quantitative PCR, full, and so on. And this is also confusing for one more reason, is that many a times RT-PCR could include reverse transcription as well, reverse transcriptase. Okay. So before going in there, I want to tell you the discussion that we had in the previous class, because I think Parvati asked a question based on that, quantifying the amount of mRNA. Say the reverse transcript, the RT-PCR, reverse transcriptase PCR, cannot tell us how many copies of RNA are present to start with. So what we can do is, or just on theoretical grounds, take 2 samples of sample A and sample B. And it is RNA sample. Assume that you have, for gene X, you have 3 copies or 4 copies. And in in sample B, you have 2 copies of gene X, mRNAs of gene X. How can you determine the quantity? How can you determine the quantity of mRNA? Or it could be of other samples also, like other nucleic acid samples. So the question is, if we did PCR, in a PCR regularly, we would have added, say normal PCR, we could have added dNTPs, we would have added buffer, DNA polymerase, and dNTPs, and we would have added water and other things to make up. We would add template. Template, assume, okay, here I said it is of RNA, so we would have to perform reverse transcriptase in the first step, and then produce the double-stranded cDNA and use it as probe. So because there are 4 molecules, we can assume there will be 4 molecules, 4 cDNA molecules. And for sample B, the same thing, everything is same. But what is different is we will have only 2 copies of cDNA, double-stranded cDNA. That is the template. Okay, C is small, okay? So now if we did first round of PCR, how many copies would have formed in each case? How many copies of gene X would have been there? You should also count the template for ease. So we had 4 copies. After one round of PCR, it would become 4 more copies, and we'll have 8 copies for gene A. I will just put it as sample A. In first round, for this, on this side we will put it as sample A, and this one here as sample B. Just the numbers, okay? Don't worry about the graph and other stuff. What would be the count of sample B? We have 2 copies initially. 2 plus 2 will become 4 copies. After second round of PCR, what would you expect? It will be 16. And for sample B, 8. And it will keep on doubling like that, right? 64, 32, and so on. Right? You can clearly see that the amount of product formed is a reflection of the template you have taken. In one case, it is 4. In one case, it is 4, and in another case, it is 2. So the product formation is a reflection of what is the template that is present in the sample, in the PCR reactions. So this is good, but we usually go till 30 cycles or so. So assume that this one, A will form 1 billion copies, and B will also form 1 billion copies. Because this for sample A, it might reach saturation very early, something like deficiency of polymerase or DNTPs or primers, something will halt the amount of product that will form, or just the kinetics also sometimes. And then at one point it will stop, and it will stop early. While when this may have stopped, for the PCR reactions for sample A might have stopped, but PCR samples for sample B will keep on going until it reaches its own stop, right? And so at the end of 30 cycles, what we will end up is that we have equal number of copies. The analysis that we do is simple: sample collection, PCR, and then electrophoresis after PCR of 30 rounds. So we are actually seeing this one. So this does not tell us the quantity of mRNA. It does not tell us, because the reactions are stopping at different times because of other reasons. So what if there is a way to be able to detect this amount of product forming in real time? What if I could check how much product has formed after cycle 1, how much product has formed cycle 2, how much product has formed after cycle 3, and so on? That is meant by real time, right? So when the PCR is happening, we should be able to estimate the amount of product that is forming. So there is one more concept we should think about before going ahead. That is, every instrument has a limitation, the limits of detection. So when you do spectrophotometry and so on, and the sample is, say, for example, I'm just making it. Sample has 2 products. Okay, 2 products. Maybe it is not within the detection limit. Say, I will put that, I'm saying that the machine can detect only when the products are about 100. Okay, and I'm going to use for sample, this is reaction 1, 2, 3, 4, 5, 6, 7, 8, 9, something like that. Each tick is a cycle number. I'll just put it to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I will go with what I have already discussed. I will try to go with this number. as it is. Okay. So I started off with sample A. Sample A had initially, it has about 4 molecules. After PCR, it will become, this is the concentrate, this is the number of products, assume that. And there is a limit. I said it can only identify, the machine can detect only if it has reached about 100 molecules. Okay. If it is below that, it cannot detect the, at least the detection is not proportionate to the concentration. So we start with, I'll use green for A. A has 4 molecules, and after round 1, it becomes 8. 8 will become 16. 16 will become 32, and about 64, and 128. And then, can you help me with the numbers? 6, 5, 256, and so on. So at which cycle is the product of sample A detected? The reading would be something like this. The machine would not be able to detect. Only here it will start detecting that part. When it reaches here somewhere, because the limit of detection is here, it will start detecting here. Okay. And then from here on, it is something like it will make it proportional. The detection is the amount absorbance or fluorescence, whatever we are seeing, is proportional to that of the concentration of the compound molecule that we are looking at. That is usually the limit of what is meant by the machine's limit. Say I would try to now talk about B. B, we said we started with 2 molecules, so it has 4, 8, 16, 32, 64, and 128 would be here. That means this detection would have started in cycle 6, and it will go on. From there on, it will be something like this. Not necessarily the same way. We will see the actual graph, how it should look like. I'm trying to only give the essence of it. So I hope you understood about the machine. I'm trying to describe here the machine. We still have a problem. How can we detect the concentration of these at all? Because we have DNA, right? DNA is forming. How can we find out how much DNA has formed? For that, we need a detection system. There are 2 popular ones. One is the CyberGreen, and other one is TACMan assays. We'll talk about CyberGreen one because it's easy, simple, and safe. It's not as accurate as TACMan ones, but it will give us good enough information. So CyberGreen is a compound that will bind to double-stranded DNA only. It does not bind to single-stranded DNA or nucleotides. So when it binds, and upon it can give also the fluorescence, and the amount of fluorescence is equal to amount of CyberGreen, and amount of CyberGreen, the fluorescing CyberGreen would be proportional to the double-stranded DNA. And after each round, we can keep looking at the fluorescence and be able to estimate the amount of double-stranded DNA. So the unbound one does not give any fluorescence. Only the bound CyberGreen will give fluorescence. So after each round, say in the previous one, if we had 16 and it gives a certain amount of fluorescence, and the machine may not be able to detect, but when it comes to somewhere here, this sample A would have given so much enough of fluorescence that the detection can be made by the machine. So that is the CyberGreen assay. In TACMan, this is ingenious and better. Nice. I think this is easier. It is a probe that is made, and probe has two things. One is a reporter dye, R is for reporter, and Q is for quencher. So there is a certain thing about the quencher and this particular reporter is that the, when they are in close proximity like this, the reporter's signal or fluorescence is quenched by this molecule. It has to do with a lot of chemistry and all, which I'm not very aware, but we know the principle of it for our purposes. So if so, the setup like this will happen. You have the DNA or say cDNA you have added to start with. You assume that you're doing your sample was RNA polymerase, you did reverse transcriptase, so you have cDNA template and you started PCR. So you will add a forward primer and a reverse primer. And then you're also going to put a probe. All of them are specific to the gene that you are looking at. Okay. The forward primer and the probe are on the same place. And when DNA polymerase takes over, and it does, our DNA polymerase will keep on adding more and more bases, and then it will come across this probe. And the DNA polymerase that we use has a 5' to 3' exonuclease activity. So as a result, it will start degrading the probe like this. When it is degraded, R is the reporter is somewhere else and the quencher is somewhere else. That means this is going to fluoresce, right? So the fluorescence of the amount of fluorescence would indicate how many of the probes are inactivated or degraded. Probe degradation is proportional to the prime cDNA, the forward primer formation, and which is indicative of the product, right? That is how you can make a link. Fluorescence is proportional to the product, and then you can estimate after each round of RT-PCR. I hope you understood that. Yes, so the DNA polymerase that you have to think about it. So normal PCR, we use stack polymerase. Okay, these polymerases may not necessarily have 5' to 3' exonuclease activity. Actually, they wouldn't usually have this. They would have 3' to 5' exonuclease activity, which is for proofreading. Okay, so yes, so you're going to use this 5' exonuclease activity. It should be present, and it is something similar to that of DNA polymerase 1, where we discussed about nick translation. I hope this is clear enough, right? So the more fluorescence is indicative of the probe formation like this. Sorry, of the product formation. Now let's look at this picture that we have discussed before. Okay, if I made any mistake in the with the diagram before, this is what you should have. Every machine has a detection limit. What you mean by detection limit is the absorbance or fluorescence here, whatever you're detecting, should be proportional to the product formed. And that is the detection limit. If it is, say, in this place, although the reading says it is, suppose, say 80, we cannot say that it is proportional. It is the amount of absorbance is not proportional to the amount of product in these places. So you call it, that is, it could be 100, 200 or so. There is a maximum limit for every machine, and that is also a detection limit. There too, it is not proportional, not. And when it is one molecule, two molecules, or four, it still probably cannot make any distinguishing difference in the amount of absorbance. That's also a limit. The most accurate one would be the midpoint of it, something like this. that would be the midpoint of detection. Okay. So where when I say midpoint, and that is it, you're saying is that is the where the absorbance is proportional to the product. Absolutely. Okay. So based on this, you can say something like this: around 22 or 23 cycles. This is called the CT value. So if you have more templates in the starting of the reaction, the CT value will be less because this is less CT value, right? It's about say 23. These are again made based on your experimental values and all. I am trying to describe the principle of RT-PCR or real-time PCR. For sample B, it is around 25, which would mean that if the CT value is more, which would mean the sample is, in the sample, the template is less. Okay. And C is very, very less. That is how you can detect it. So there are several systems where I think we can watch some videos on how RT-PCR works. It is after each round of PCR, say it can be done in a 96 well microtiter plate. They are special plates that are made. And after each round, the machine will check how much of fluorescence is present in each of the wells. And after first round, it will make readings. After second round, it will make readings, and third, and so on. So that is how the machine will read after each round of PCR. And then plot a graph like this, based on which we can interpret that sample A had, because it's detected earlier and so on, sample A had more template, and sample C had the least of the templates. Okay, that is RT-PCR. Okay, I'll stop here. And if you have any questions, I will try to answer. Do you have any questions with the question that we have discussed, this question? Anybody has any questions? Or if you have found any other answers, you can also say. Some people have used linkers, and some people have used exonucleases and so on. So if you have alternate ways, you can tell me, and we can still consider it. But ensure, what you should ensure in this case is that, say, for example, I have treated it with KPN1 or so. I mean, I'm just trying to discuss precautions only. If I have something like this, assume that this is the 5' end, and this is 5' end. So you have 3' OH. If I use exonuclease, that is 5' to 3' exonuclease, it will cut. It will cut on this side, and there is also it will also cut on this side. That is one thing you have to remember. If I'm using somehow, if I'm trying to use a Kleinow fragment and fill it up, filling up will happen on both the sides. You have to think about, also keep in mind, anytime whatever modifications you're doing here, and the required modifications, it should not have happened in this place. So that you should keep in mind. Okay. Yes. Any questions? Anybody?
Sir, this is based on the solution that you have suggested.
Yes.
So you mentioned that we can use both P and M to digest the expression vector. Like, is there a necessity for us to use two different restriction enzymes there? Because there's no need for us to replace it, right, sir? We can just insert it.
Say it again, please. I missed it. Yes. Tell me once.
So
So I
you
want it to
said,
be in this direction, right? That way, that is clear?
yes, sir.
Yes. Okay. Tell me.
So you said we would use P and M. So the part which contains T will be out, and then we'll be replacing that particular part of the DNA with our gene of interest.
Okay. Yes.
But why
Yes.
not use a single enzyme? It will have similar overhangs, and we could just go with that, no, sir. Like, is there
Yes.
a
Which one?
size-based?
You tell me which one is the. I will erase all. Oh, sorry. Mm, tell me what. Uh, tell me what exactly do you want to do? We'll go. We have 4 minutes. We can go along what you're saying. Yes. Tell me the series, and then we can probably go understand it easily. It will be useful for others too. Okay. Tell me what do you want to do with me with this cloning vector? What should we do?
So, as per your suggestion, use B and A.
Okay. B and A. Okay, this is B and A. This will be B overhang, and here is A overhang. And A is a blunt end, and B is not. B is a 5' overhang. Yes, now go ahead.
For the expression vector, use only M.
Only M.
Yes, sir.
Okay, if we use only M, what would you get? What is for M? M is a 5' overhang. Here we have a 5' overhang. Here we have 5' overhang. For the fragment, we have 5' overhang on the B, right? And on the A side, we have a blunt end. How would it fit in here?
So why not use the clino fragment again?
Yes, I know. Clino fragment, and what do you do with that? Clino fragment where? To which one?
After mixing and ligating, because those have
No, no, no.
compatible over.
Do you want to
Oh,
do the with the fragment, or which one do you want to use the Clino fragment for? I will remove this. Okay, we don't need this anymore. Yes, tell me. Clino fragment for which one? So
so I
what
guess
would
this
we have?
strategy does not work because
Yeah,
both of them have 5' overhang. So
GTC. Now, if this is a blunt end on this side. Now, which one do you want? We can try. When we make mistakes, we are on the right path. We'll get to when we identify mistakes, we know what will be the right one. So, which one do
I'll
you want
use
to
it
treat
for
it with
the
Clino fragment?
vector expression vector.
Do you want to treat this with expression vector
But it
with the
won't
Clino
work, so because
fragment?
both
Both
of
will
them
become
has that
blunt ends.
problem. Yeah.
Both will become blunt, right? We can also make both of them blunt here too, but then we will get 50% of them in proper the desired one and 50% in the reverse one, and then we'll have to again do a screening to identify which one is in the correct orientation and then go about it. It is a strategy, yes, certainly. But the question is, I think I have given this almost the same question in as a class as well as a homework also. So, but that is how we learn, right? Not every time we look at. When I'm framing questions, I have to take a lot of care that other possibilities are not that easily. So what we know from this is to look for isocodamers. That is the easiest one, because you just restriction dilution ligation, you don't have other treatments. What we should also keep in mind, I'll stop with this, don't so if we have more number of steps, step one, digestion, then we have to do electrophoresis and purification. Step two, treat with clinofragment. Again, we have to do electrophoresis and purification. Step two, again, after ligation, we don't have purification. After each round of electrophoresis and purification, we will be losing the DNA. The concentration of DNA that at the end that we will have for ligation will become lesser and lesser. And that's one of the reasons why we should try to do something in minimum number of steps. It is better, it is less time consuming, less resource consuming, and it is easier that way. Okay.
Thanks a lot for the
Yes.
clarification, sir.
Yes, good. Any other questions, please? Okay, Prashanti, Ramya, Prahladan, Sajit, Parvati, Yogeshwaran,